Welcome! This is the post-translational modification video for BS 3336 Undergraduate Advanced Experimental Biology UAEB, presented by Teo Zhi Ting and Shuhan. A post-translational modification, PTM, is defined as any modification of a protein structure that occurs after synthesis of the protein via translation of the messenger RNA. Every eukaryotic protein is known to undergo proteolytic processing and modifications of functional groups of selected amino acids. The side chains of non-polar aliphatic amino acid and of phenylalanine do not participate in PTM. But active groups such as an amine, thioether, carboxylic acid, and hydroxyl are susceptible to covalent modifications in a biological milieu. More than 300 PTMs are known. The best characterized PTMs in eukaryotes are phosphorylation and glycosylation, but also include other common PTMs such as acetylation, methylation, ubiquitination, simolylation, and citrullination. Often, these modifications have a significant influence on the activity and specificity of proteins. Also, they might play a role in stabilizing the protein structure and regulating the enzyme activity. Therefore, the characterization of PTMs has attracted much attention. However, the full extent and functional importance of protein modifications in the working cell are not well understood because of a lack of suitable methods for their large-scale study. A single protein can be modified not only at multiple sites within the molecule, but also in various combinations of those PTMs. In addition, protein modifications at given sites are typically not homogeneous. A specific modification may occur only partially at a given site of the protein. The amount of protein in a single modification state can thus be a very small fraction of the total amount of the whole population of the protein. Thus, the specification of a PTM at a given site requires in general a large amount of protein. Furthermore, a single gene can give rise to a number of gene products as a result of alternative splicing. All of those contribute to the extreme complexity of an entire proteome and cause difficulty in characterizing even a specific type of PTM on the proteomic scale. In this video, we will focus on a type of enzymatic post-translational modification called citrullination. Citrullination or deamination is the conversion of the amino acid arginine in a protein into the amino acid citrulline. Citrulline is not one of the 20 standard amino acids encoded by DNA in the genetic code. Instead, it is the result of a post-translational modification. Citrullination is distinct from the formation of the free amino acid citrulline as part of the urea cycle or as a byproduct of enzymes of the nitric oxide synthase family. Peptide-bound arginine residues can undergo post-translational modification, and the resulting citrulline remains part of the protein as peptidocitrulline. The conversion of arginine to citrulline changes the charge of the amino acid. arginine is positively charged at a neutral pH due to the presence of a guanidino group that could easily be protonated at physiological pH. The removal of the imino moiety is an enzymatic reaction catalyzed by peptidoarginine deaminases, PADS. The resulting citrulline lacks the strong basic character. It is a neutral amino acid similar to asparagine or glutamine. The protein deamination process is catalyzed by a family of peptidoarginine deaminases, PAD1 to 4 and PAD6. A key regulator of PAD enzymes is calcium. Binding of calcium ions activates PADs. Under physiological conditions, the calcium concentration is low. Therefore, PADs are inactive. This suggests that citrullination occurs only under extreme conditions, such as during apoptosis or terminal differentiation of the epidermis. However, PADs are implicated in other cellular processes, such as gene regulation, which will be mentioned in the later section. In 2004, the catalytic mechanism for peptidoarginine deaminases was proposed. The active site of the enzyme contains cysteine residue that can attack the guanidino group of the arginine and establishes a tetrahedral adduct transition state intermediate while ammonia is released. The adduct is then cleaved by the nucleophilic attack of a water molecule. This latter step regenerates the cysteine residue and results in the formation of a keto group as well as a new primary amine. The deamination process could change the primary, secondary, and tertiary structures of proteins. In general, citrullination alters the protein structure and results in a somewhat looser, less organized, more open configuration, which will link to the section we would like to discuss next: roles of citrullination in both physiological and pathological areas. Now, let's understand how citrullination plays an important role in the physiological area. In non-apoptotic cells, citrullination involves in maintaining and organizing keratin matrix at the epidermis terminal differentiation stage. Before differentiation, filaggrin is stored as profilaggrin. During terminal differentiation, profilaggrin is citrullinated at the beta turns of the protein, allowing it to adapt to a more open and disordered structure that make it susceptible to proteolysis. Calpain then digests the citrullinated protein into small peptides that are used to cross-link keratin filaments into an organized matrix. Another role of citrullination is the regulation of genes. PAD4 is localized in the nucleus in granulocytes, and it can deactivate genes by deaminating histones such as H2A, H3, and H4. For instance, estrogen-dependent genes are induced by histone methylation. When specific histone arginine residues are methylated, chemical attractions between histone and DNA is weakened, allowing DNA to uncoil from the nucleosome so that transcription factor proteins and RNA polymerase can access the DNA. PAD4 can demethylate these histones by converting the methylated arginine to citrulline, hence reversing the transcriptional activation by histone methylation. In addition, deamination of arginine will produce citrulline that can no longer be methylated. However, it is important to note that arginines that are dimethylated cannot be deaminated by P84. Citrullination plays an important role in nuclear laminar disassembly in apoptosis. During apoptosis, intranuclear calcium ion concentration increases and activates PAD enzymes. The activated PAD then deaminates a 70 kilodalton protein that is attached to the nuclear lamina. This reduces the isoelectric point of the protein and causes it to dissociate from the phosphorylated serine residues at the nuclear lamina. This series of events leads to the disintegration of the nuclear lamina during programmed cell death. After the disorganization of the nuclear lamina, DNA can be released into the cytoplasm and degraded by nucleases. Another apoptosis-related role of citrullination is to disassemble vimentin filaments. Vimentin is an intermediate filament expressed by many cell types, which is important for maintaining the shape of the cell and the organization of organelles. They are very stable and are less likely to undergo constant changes during the cell's lifespan. During apoptosis, the high intracellular calcium concentration activates PAD, which deaminates and reduces the isoelectric point of the head domain of vimentin. As a result, soluble vimentin is unable to polymerize and filamentous vimentin undergoes depolymerization. Therefore, the vimentin intermediate filament network is lost and the cell undergoes huge morphological changes. The last apoptosis-related role of citrullination is to disassemble nucleosome. During apoptosis, the high intranuclear calcium ion concentration allows P84 to attain its maximal activity. The extremely active P84 deaminates histones non-specifically, which will greatly reduce their positive charges. Without their basic arginine residues, the binding strength of histones to the acidic DNA is reduced. As the nucleosome to unfold, DNA becomes susceptible to nuclease attack. After presenting physiological roles of citrullination, Shuhan will be presenting pathological roles of this type of post-translational modification.
The first pathological consequence of citrullination that I will be discussing is rheumatoid arthritis, or RA in short. RA is a chronic autoimmune disease which causes inflammation and swelling of the joints. If it is not treated, it will lead to the permanent damage of the joints or even result in disability. About 1% of the adult population is affected, and women are 3 times as likely to be affected as men. The serum and synovial fluid of RA patients contain high concentration of autoantibodies against various targets, not all of which are specific to the disease. However, 2 autoantibody families are found in most RA patients. One is the rheumatoid factor, or RF. The other is the family of anti-citrullinated protein antibodies, or ACPA, which recognizes the deaminated alpha and beta chains of fibrin. Because ACPA is highly sensitive and highly specific to RA, it is currently the best biomarker for the disease. Let's discuss the first factor that causes RA. It has been suggested that fibrin plaques found in synovial tissues is a possible cause of RA. Under normal circumstances, small amounts of fibrinogen and other procoagulant proteins can pass through the capillary wall and travel to the interstitium, where they are broken down into fibrin peptides and fibrin monomers, which can then polymerize to form plaques. These plaques are subsequently degraded by enzymes such as the serine protease plasmin, or may be taken up by macrophages. However, in RA, the elimination of polymerized fibrins reduces the number of cleavage sites of plasmin, leading to the accumulation of polymer fibrin and the accumulation into fibrin plaques. Fibrinogen itself can activate innate immunity by stimulating the CD11C-CD18 complex, toll-like receptor 4, and CD14 on the macrophages. These immune cells then produce various pro-inflammatory cytokines like IL-1, IL-8, IL-12, and TNF-alpha, causing inflammation in the synovium. On the other hand, as citrulline is not one of the 20 amino acids encoded by our genetic code, citrullinated alpha and beta chains of fibrin are immunogenic and can be recognized by ACPA, thus activating the adaptive immunity and causing inflammation. Several studies suggest that environmental factors such as smoking, vibration, exposure to mineral dust, and injury increase the risk of developing RA. Under these circumstances, which induce cell necrosis, the integrity of the cell membrane is lost and the cytoplasm containing enzymes like PAD is released from the cell. High interstitial concentration of calcium ions will activate PAD to deaminate proteins such as vimentin. The peptide of these proteins can then be presented to T cells by antigen-presenting cells, inducing immune response. In addition to environmental factors, some people are genetically predisposed to RA. They have MHC class II molecules which contain a positively charged motif, glutamine lysine arginine alanine alanine, or glutamine arginine arginine alanine alanine in the HLA-DR beta chain. This motif is in the P4 pocket of the MHC class II molecule. As arginine is a positively charged residue, arginine-containing peptides bind unfavorably to the MHC due to the electrostatic repulsion between the peptide and the MHC. On the other hand, peptides containing the neutral citrulline can bind to the MHC with a much higher affinity, hence inducing an immune response. The next pathological consequence of citrullination is multiple sclerosis, or MS in short. MS is an immune-mediated disease in which the body's immune system directs an abnormal attack against myelin and nerve fibers of the central nervous system. When the myelin shaft or nerve fiber is damaged, the transmission of nerve impulses is disrupted, leading to a wide range of symptoms. This disease will eventually lead to paralysis and death. So how does citrullination lead to MS? Research has shown that MS is primarily caused by over-citrullination of myelin basic protein, or MBP. In myelin shaft, MBP helps to pack lipid bilayers together through the ionic interaction between the MBP's positively charged arginine residues and the negatively charged phosphatidylserine in the lipid bilayer. This makes the myelin shafts tight and compact. Citrullinated MBP loses its basic residues. Hence, its interaction with the negatively charged phosphatidylserine is weakened. As a result, the myelin sheath loses its compactness. Citrullinated MBP is also more susceptible to degradation by the metalloproteinase cathepsin D. Deamination opens up the structure of MBP and exposes its buried phenylalanine-phenylalanine linkages to cathepsin D, causing citrullinated MBP to be degraded 5 times faster than its native form. The released phenylalanine 14 phenylalanine 88 peptide will induce an autoimmune response. As a result, immune cells will attack the nervous system, causing local inflammation and the destruction of the nerve cell and myelin sheath. Research has also suggested that the increase in citrullination of MBP in MS is a result of reduced methyltransferase activity. Where MBP is not methylated, the protein has a lower hydrophobicity and adopts a less condensed structure. Hence, the unmethylated protein is more prone to citrullination. The last disease that I'm going to talk about is Alzheimer's disease, or AD in short. AD is a chronic neurodegenerative disease that develops slowly and gets worsened over time. In advanced Alzheimer's disease, brain cells such as neurons and synapses are massively lost. Surface area of the cortex and sizes of hippocampus and ventricles of the brain will be greatly affected. These morphological changes will lead to difficulty in thinking, planning, and remembering, and eventually cause the patients to lose ability to carry on a conversation and respond to their environment. Although there's no cure for AD currently, effort of AD research such as looking for potential AD biomarker has been placed on diagnosing the condition before symptoms begin. Citrullination can serve as a potential biomarker for Alzheimer's disease. In neurodegeneration of type 2 astrocytes, the PAD2 mRNA production and translation increase. At the same time, the increase in the calcium ion concentration will activate PAD2, causing it to over-citrulinate GFAP. GFAP, which stands for glial-fibrillary acidic protein, is a major intermediate filament in astrocytes. Although the physiological role of GFAP deamination is still unknown, it is found that the expression and deamination of GFAP are increased when neurodegeneration is induced in astrocytes. As GFAP deamination is characteristic for Alzheimer's disease, citrullinated GFAP could be an early marker in neurodegenerative disease. So, what are the future directions for research in citrullination? Researchers have suggested a two-level model for citrullination. At very high calcium ion concentration, PAD is fully active and it has broad substrate specificity. This means that it can deaminate any arginine-containing protein. This could lead to autoimmune response as novel citrullinated protein will be recognized as antigen and targeted by the immune system. On the other hand, at low calcium ion concentration, PAD activity is regulated and it has a higher substrate specificity, for example, in gene regulation. However, more research needs to be done to identify the specific substrates of PAD and elucidate the mechanism of PAD activity at low calcium concentration. Another interesting direction of research is to find out if there are any pathway that allows citrulline in deaminated protein to be converted back to arginine. For instance, in gene regulation, it is unlikely that citrullinated histones are degraded and new arginine-containing histones need to be synthesized to replace them. Perhaps a peptidyl citrulline imminase is waiting to be discovered. We have two files for citrullination analysis. Each one comes from a different human heart plaque sample. We will identify which proteins are citrullinated in the two samples and compare the difference between the two samples. From the generated CSV output file, we see a huge list of sequenced peptides. The headings for the columns include protein description, which is the FASTA title or description line of the protein, protein score, protein mass, peptide expected MR, which is the observed relative molecular mass of the peptide, peptide expected Z, which is the observed charge state of the peptide, peptide calculated MR, which is the calculated relative molecular mass of the peptide, peptide delta, which is the calculated relative molecular mass minus the observed relative molecular mass, peptide start, the starting residue position for the peptide in the protein, peptide end, the ending residue position for the peptide in the protein, peptide miss, number of missed enzyme cleavage sites, peptide score, peptide match score or the peptide ion score, peptide homology, which is the peptide score homology threshold, peptide identity, which is the peptide score identity threshold, peptide expect, which is the peptide match expectation value, peptide sequence, peptide variable modifications such as citrullination, oxidation, peptide variable modification position, which is the position of the modification along the sequence of the peptide. And finally, peptide non-match, number of fragment ion matches used for scoring. Now, you may wonder, what is the peptide identity threshold and what is the peptide homology threshold? In MASCOT, the ion score for a tandem MS match is calculated from the probability P that the observed match between the experimental data and the database sequence is a random event. The peptide identity threshold is set at a 0.05 significance level. This means that if a peptide score is greater than the identity threshold, it has a less than 5% chance of being a false positive. However, if the quality of a tandem MS spectrum is poor, especially when the signal-to-noise ratio is low, a match may still be correct even though the score does not exceed its identity threshold. MASCOT can identify such a match when its score is sufficiently larger than the scores of the other random matches. Based on the distribution of random matches, MASCOT provides a second lower threshold to single out possibly correct matches that are lower than the identity threshold. This second threshold is called the homology threshold. In each CSV file, we created another two columns. One is called greater than homology threshold, which is peptide match score minus the peptide score homology threshold. The other is called greater than identity threshold, which is peptide match score minus the peptide score identity threshold. Then we sort the data first by peptide variable modification, then by greater than homology threshold. We extracted the rows of peptides which have citrullination variable modifications and have a peptide score greater than the homology threshold. Then, from this new data, we sort the data first by the protein accession number, then by greater than identity threshold. Here you see the list of citrullinated peptides. The bolded ones have peptide score greater than identity threshold, while the non-bolded ones have peptide score lower than identity threshold but greater than homology threshold. We then counted the number of citrullinated peptides for each protein and then plotted the data using a bar graph. The blue bar shows the number of peptides that have scores greater than the identity threshold, while the combined blue and orange bar shows the number of peptides with scores higher than the homology threshold. From the bar graph of the citrullination 1 file, you can see that vimentin, transgenic, Ig-alpha-1 chain C region, and Ig-alpha-2 chain C region have the most number of citrullinated peptides. From the bar graph of the citrullination 2 file, you can see that fibronectin, collagen alpha 2 1 chain, serine protease HTRA1, and hemoglobin subunit beta have the most number of citrullinated peptides. Previously, we have discussed that citrullination of vimentin plays a role in apoptosis. Perhaps the high level of vimentin citrullination in the first sample is a result of apoptosis. Other biomarkers for apoptosis, such as cytochrome C or cytokeratin, can help us confirm if apoptosis is indeed taking place in the first sample. We acknowledge that our treatment of the data is crude. It only gives us a rough idea of which proteins are citrullinated, but it does not allow us to accurately quantify the relative amount of citrullinated proteins in the two samples. This is because peak intensities can vary between runs due to variation in LC-MS-MS condition. In order to do a relative quantification, the two samples can be labeled by two sets of isobaric tags, such as iTrack. The samples are then mixed and run together in a single LC-MS-MS run. Because the two tags have the same mass, peptides carrying the same post-translational modification can be eluted and sequenced in tandem MS together. During tandem MS, the reported group of the isobaric tags are generated due to fragmentation. The quantification of the peak areas of the reporter groups can give us a relative amount of a citrullinated peptide from a given sample. Finally, I and Zhi Ting would like to express our thanks to Professor Sze and the TAs.